Hi guys. Whoops. All right. I hope this gets to go up on the internet soon because for some reason my internet's not working right now. I'm going to go through this lecture pretty fast. You guys should know this stuff already, but I want to focus on some things like polarity, polar bonds, and a few other concepts that I, uh, excuse me, that I've had experience with. People don't tend to get them as easily, and I want to make sure you have a good foundation in these basic biochemistry issues, because these are really going to form the foundation for the rest of what we do for the rest of the semester. We are going to come back to polar bonds and hydrophobic interactions and ionic interactions for the rest of the semester. We're going to be talking about the specifics of different proteins for the rest of the semester. So you really, really need to have a good foundation in this stuff. This lecture can be found on our website. The PowerPoint or the PDF of the PowerPoint can be found on our website under lectures. So I'm not going to talk too much about covalent bonds. You guys should already know, that a covalent bond is a bond between molecules, between atoms that are sharing electrons. What I want to focus on are polar covalent bonds. And when you have a polar covalent bond, that means that the electrons within that covalent bond are not being shared equally. And this is due to differences, strong differences in the electronegativities between those atoms. And the result of polar bonds is that you have partial charges. So the atom that retains more than its fair share of the electrons, so the atom where the electrons spend most of their time, has a partially negative charge associated with it. In the case of water, that would be the oxygen, which is much more electronegative than hydrogen. And then hydrogen does not have as much of the electrons surrounding it, and so hydrogen is associated with a slightly positive charge. And because you have these partial charges on the molecules, you have electrostatic attractions. So partial positive charges are attracted to partial negative or even fully ionized negative charges. And this is the basis of hydrogen bonding. Okay, so electronegativity is a relative measurement of how strongly different atoms pull electrons towards themselves. So they're not, the electronegativity, the scale isn't associated with any units, but you can use those measures to compare one atom with another. So you can tell, is there a large difference between the two atoms? So will this form a polar bond, or is there a small difference in electronegativities between the two atoms? So there won't be such an unequal sharing of the electrons. And so here, down at the bottom of this slide, you can see the relative electronegativities between hydrogen, carbon, nitrogen, and oxygen. And one of the most polar bonds that you see in water and other alcohols is the OH bond. And so you can see that the oxygen has a high electronegativity of around 3.5, whereas hydrogen has a low electronegativity at about 2.2. So this difference between the electronegativity of the oxygen and the electronegativity of the hydrogen cause the electrons to be more attracted to this oxygen. And so the electrons, being more attracted to the oxygen, are going to spend more of their time around the oxygen atom. So this is going to cause a more negative charge associated with that oxygen, whereas the hydrogen is not attracting those electrons strongly, and so the oxygen pulls the electrons away from the hydrogen, which means that the hydrogen has less of a negative charge, and so it has a partially positive charge associated with it. Now, some people mistakenly want to think that if an atom is highly electronegative, it will always be a polar bond. But you have to remember that whether a bond is polar has to do with the difference of electronegativities. So for example, in molecular oxygen, both of these oxygen atoms are highly electronegative. But this oxygen atom is just as electronegative as this oxygen atom, so that the electrons are going to be shared exactly equally. There's not going to be more of a force from one oxygen than from the other because they have the same electronegativity. There's a difference of 0. Okay, so this is not a polar bond. Okay, the molecular oxygen does not have a polar bond. All right, so I'd like you to go through these different bonds and ask yourself, is this going to be a polar covalent bond, a slightly polar covalent bond, or a non-polar covalent bond? And I want you to appreciate that this is a spectrum, okay? So this goes from having, when the electronegativity difference is high enough, then you get ionization, right? So between something like chlorine and sodium, you have such a large difference that the chlorine atom is able to completely remove the electron from the sodium atom. When you have a high difference that's not quite that high, such as in the OH bond, you have a highly polar covalent bond. Okay? When you have something like carbon and nitrogen together, you have a slightly polar covalent bond. When you have something like a carbon and hydrogen together, you have a nonpolar covalent bond, even though there's still some difference there. So I want you to go through these bonds, the carbon-carbon bond, the carbon-hydrogen bond, the OH bond, the NH bond, the CN bond, the CO bond, and look at them and see how there is a spectrum of polarities. And so you'll have more of a negative charge associated with an oxygen in an OH bond than you would in a CO bond. And so I want you to kind of appreciate that spectrum. All right, so a polar bond is a polar covalent bond. This is a bond between two atoms in the same molecule. Okay, this is intramolecular bonding, the polar covalent bond. Polar covalent bonds lead to hydrogen bonding. A hydrogen bond exists when you have an electrostatic attraction between partial positive charges. This is an intermolecular bond. Hydrogen bonds are intermolecular bonds because they occur between two different molecules. Hydrogen bonds do not create a molecule. Covalent bonds create molecules between atoms. Hydrogen bonds stick two molecules together transiently. Okay, hydrogen bonds are not very strong bonds, but collectively they can cause great forces to occur. So if you look at this network here, you have a water molecule in the center, and the hydrogen has a partial positive charge associated with it. That partial positive charge is going to be attracted to the partial negative charge on the oxygen of another water molecule. This is going to stick those two water molecules together. Right, so you have a cohesive force. So you can pour water easily, or water can travel up the trunk of a tree from the roots to the leaves because of this hydrogen bonding, sticking those water molecules together. These hydrogen bonds are also really important in protein folding, and we'll see, and also, other higher molecular forces. So the DNA secondary structure. The hydrogen bonds are responsible for holding the double helix together between purines and pyrimidines. So hydrogen bonds are an important molecular force, but remember that they do not create molecules. They only stick molecules together. So water forms a highly organized network because of its ability to hydrogen bond. And when you have many water molecules together, they form this network because each water molecule can form 4 hydrogen bonds. Each oxygen can bond to 2 other hydrogens, and each hydrogen can bond with 1 oxygen. Right? This is really important because water, because of these hydrogen bonding properties, make life possible on Earth. Before we get to hydrophobic interactions, I'd like you to pause for a moment and I'd like you to write this down. There are properties of water that are due to hydrogen bonding or to their polar nature. And I want you to think about these properties: adhesion and cohesion, surface tension, high specific heat, and what's the other one? And its solubility. Okay, water is a good solvent. So that's adhesion, cohesion, high surface tension, high specific heat, and it's a universal solvent. And I want you to write down, how does hydrogen bonding or having strong polar covalent bonds, specifically, how does the hydrogen bond or the polar bond lead to each of these properties? Okay, I want you to be explicit about the hydrogen bond or the polarity and how that leads to these 5 different characteristics of water. Okay, so just take a minute now. You can visit your textbook or other websites, I don't mind. But I want you to write that down and turn that in the first week of December. All right, so hopefully you've paused and taken care of that, and then come back to this lecture. I want to spend a little bit of time on hydrophobic interactions. We'll be talking about hydrophobic interactions, especially in terms of protein folding and membrane chemistry. Hydrophobic interactions, the name is actually a bit of a misnomer because it's not an attraction between hydrophobic molecules. The attraction is actually due to the fact that these hydrophobic molecules are being repelled by the water. Right? So if you have hydrophobic molecules inside of a hydrophobic environment, they're not going to be attracted to each other. But once you put hydrophobic molecules inside a hydrophilic environment, inside an environment where you have polar bonding occurring, for instance, in an aqueous environment, a watery environment, like in the cell, the hydrophobic or the non-polar molecules are going to be pushed together because all of those polar molecules, like water, are looking to make hydrogen bonds. And so their most energetically favorable state is having their unlike charges stuck together, right? So the partial positives are going to be stuck to a partial negative. When they find something that has no charge associated to it, that isn't attractive to them. And so those uncharged, completely uncharged molecules are going to be pushed to an area that minimizes their surface area. It minimizes their exposure to the polar molecules. And so it's really the hydrophilic molecules that stick the hydrophobic molecules together. It is not an attraction of the hydrophobic molecules innately. It's not innate to the hydrophobic molecules. It's a function of the hydrophilic molecules looking for other hydrophilic molecules so that they can form hydrogen bonds. In order to reduce the surface area, the hydrophobic molecules tend to aggregate together. All right, and then this is a way for you to not forget that polar molecules result in hydrophilic molecules, right? Why do white bears like to go swimming? Because they're polar bears. All right, I'm not going to spend too much time on ionization, but you should know what ionic bond is. I do want to point out that in an aqueous environment, you have very few ionic bonds. And what we have instead in biological systems, which are aqueous systems, are ionic interactions. You have the attraction of the negative to the positive. But because in an aqueous environment, you always have the partial charge of the hydrogen, the partial positive of the hydrogen is always going to be attracted to that full charge of the negative ion, and the partial negative of the oxygen in the water will always be attracted to the fully positive charge of your cation. And so this is going to form what's called a hydration shell around those ions. And so because you have water surrounding those ions, they are not able to form those strong crystalline structures, those strong ionic bonds that you see in dry chemistry. Okay? Cell biology is aqueous chemistry. We're in a wet, watery environment. And so unless you're in the heart of a protein where water is excluded, you don't get ionic bonds. All you get are ionic interactions. And ionic interactions are strong, but they're not molecular forces. They don't form molecules. Okay, they're not like a covalent bond. They're stronger than a hydrogen bond, but not as strong as an ionic bond. All right, you should know dehydration or condensation reactions, which are reactions that build molecules. Dehydration and condensation reactions, those are synonyms. They require energy. They build complex molecules from smaller, less complex molecules. And the opposite are hydrolysis reactions. Hydrolysis reactions break apart complex molecules into smaller molecules, and they produce energy. Okay, so this is the difference between anabolic reactions, which build molecules, and catabolic reactions, which break apart molecules. All right, you should go through these. All right. So hopefully you can answer all of those questions. Just remember, if you're building a molecule, if you're building a larger molecule from smaller pieces, it's a dehydration reaction. And if you're breaking a molecule apart or you're making smaller molecules from a larger molecule, it's a hydrolysis reaction. Okay? Hydrolysis reactions produce energy, dehydration reactions require energy. All right, we are going to focus on proteins for the rest of the semester. Everything we do, we're going to explore the proteins that are responsible for what is happening. And so you guys need to have a really good foundation in the vocabulary and the basic molecular forces that are happening, right? I'm not going to spend a lot of time on this, but I do expect you to know the structure of an amino acid, right? You have your central carbon, your amino group, your carboxylic acid group, and your R group. You should be able to recognize an amino acid if I give you a group of molecules. The R group is responsible for the properties of those amino acids. And so you can group R groups in different ways. You have nonpolar R groups or hydrophobic R groups. Notice these have primarily CH bonds because the CH bond is nonpolar. So if I say which of these is a nonpolar amino acid, you would look for the nonpolar R groups. These are polar R groups. Notice most of these have the OH bond. OH, OH, this has an SH bond. Sulfur is also highly electronegative. Here you have the CO bond, right? Proline is unique because it includes the amino group in the R group because it forms this ring. So that's kind of an oddball. I wouldn't ask you that. You have charged R groups. These are the positively charged R groups. You can see the positive charge on the amine down here. These are the negatively charged R groups. They're carboxylic acids. Sometimes you see these instead of as aspartate or glutamate, you'll see these as aspartic acid or glutamic acid. Okay, so just be aware of that. If this has the H attached to it, if it's not ionized, it'll be known as aspartic acid or glutamic acid. These are basically the same molecule, it's just at a different pH. So if it's aspartate, it's ionized, and if it's aspartic acid, it's got the acid still attached. It's got the proton still attached. Okay, the aromatic R groups have rings. Tryptophan, tyrosine, phenylalanine. They all have those benzene rings. All right, so which of these is hydrophobic? What are you going to look for? You're going to look for the hydrophobic R group. Remember, you'll always have the amino group and you'll always have the carboxylic acid group, and those are never going to be hydrophobic, but that's not what you're looking for. You're looking for the hydrophobic R group. All right? All right, so again, you should know the 4 levels of protein structure. The primary structure involves covalent bonding, the peptide bonds that determine the sequence of amino acids. And it's this primary structure that is going to dictate all of the higher levels of structure because it's the interaction between those amino acids that are going to determine how they fold, right? If you have lots of hydrophobic amino acids, those are all going to nestle inside the center of an aqueous protein. Or if you have a lot of hydrophobic amino acids, it might form an alpha helix that then becomes embedded in the membrane. Okay, so the sequence of amino acids will determine how it folds. The secondary structure involves the hydrogen bonding in the backbone of the protein. Okay, the backbone of the protein, I'm gonna back up for a second to our amino acid here, sorry. This is the backbone of the protein. If you look at the amino acids, so this amino group is going to attach to the carboxylic acid group of the previous amino acid. And so you get a chain, and the backbone goes NCC, NCC. Amino, central carbon, carboxylic acid. Amino, central carbon, carboxylic acid. That's the backbone. And then sticking out from the backbone, you have all of these R groups. All right, going forward again, when you talk about the secondary structure, you're talking about what's happening in the backbone. How is the backbone stabilizing itself? And there are 2 basic motifs that the backbone will commonly engage in. Not always, but often. You have the alpha helix, where a helical form is assumed, and you have hydrogen bonding between a hydrogen and an amino group with the oxygen and the carboxylic acid. Okay, we'll talk more about this in a second. In a beta-pleated sheet, instead of taking on a helical configuration, you get these sheets. And sometimes it's contiguous pieces of the protein where they're all right next to each other, but sometimes it's not. Sometimes you'll get a piece of protein and then another piece will go do some alpha helix or a hairpin loop, and then it'll come back and you'll get another piece that forms the beta-pleated sheet with it. So beta-pleated sheets are a little bit flexible, but they always involve hydrogen bonds between the oxygen on a carboxylic acid and the hydrogen on an amino group. And that's going to be in neighboring portions of the beta-pleated sheet. Okay. Now, not all of the protein will take on one of these motifs. And I'll show you a picture of that in a little bit. But first I want to talk more about the alpha helix. Okay, so the alpha helix is a very structurally stable configuration. So you see alpha helix a lot in structural proteins like collagen. You see alpha helixes in membrane spanning domains. And you usually see multiple alpha helices in many, many, many proteins. Okay, so what the alpha helix does, it stacks the backbone on top of each other. Okay, so if you look at this, this is the alpha helix looking down the helix. And the backbone of the helix is all right here, all twisted up, with the R group sticking out of the helix. And this allows the chemistry in this part of the protein to be dictated by the R groups. Remember, the backbone, the amino groups in the carboxylic acids, those are charged or at least polar, depending on the pH, groups. And the protein might not want to be reacting with the environment of the cell. By putting them in this configuration, you're neutralizing those polar groups by allowing them to hydrogen bond with themselves. So here you have this polar oxygen hydrogen bonding with this polar hydrogen. Now those parts of the molecule aren't going to want a hydrogen bond with, for instance, water or other proteins that are in the cytosol. So you are removing the chemistry of the backbone from play, and you're allowing only the R groups to be the part that interacts with the environment. All right, a similar thing happens in the beta-pleated sheets. So here you can see the amino group is hydrogen bonding with the carboxylic acid group. Okay, now this amino group isn't going to want to hydrogen bond with the oxygen of water. This oxygen isn't going to want a hydrogen bond with the hydrogen of water. They are being neutralized because they're hydrogen bonding with themselves, and so now these R groups are free to interact with the environment. All right, I want you to take a look at this 3D model of a protein, and I want you to notice that there are multiple alpha helices in a single protein. Now, some people think the whole protein has to take on either an alpha helix or a beta-pleated sheet. But notice here that you have an alpha helix here, and you have an alpha helix here, and you have an alpha helix here, and here, and here, and here. And you have multiple beta-pleated sheets as well. These arrows represent the strands of the beta-pleated sheet. So you have a beta sheet here and here. You have some beta sheet there. And again, you have some here and here, and one going in a different direction there. So you have multiple secondary motifs in a single protein. Notice these little squiggly lines. Those are representing regions that aren't conforming to either the alpha helix or the beta-pleated sheet. Okay, so that's possible too. All right, so when we talk about the tertiary structure of the protein, we're looking at how is the protein folding up and taking on a 3D configuration. the tertiary structure is going to be affected by all sorts of different chemical interactions. Okay, so the primary structure was determined by covalent bonding. The secondary structure was determined by hydrogen bonding. The tertiary structure involves everything: hydrogen bonds, hydrophobic interactions, van der Waals forces, which have to do with nonpolar attractions, ionic interactions, sometimes covalent bondings. That's a little bit more rare and usually happens in secretory proteins, proteins that are going to be excreted outside of the cell. You get these disulfide bridges, and that's going to make it so that even when the protein goes into an uncontrolled environment, it will still retain its proper configuration. Okay. Now, some proteins only have tertiary structure. Proteins that consist of a single polypeptide will only have tertiary structure. But most proteins consist of multiple polypeptides or multiple subunits. And so those types of proteins will have quaternary structure. Now, actually, quaternary structure can arise in two ways. One of the ways is by having multiple polypeptides. Okay, so an example of that is collagen. Collagen consists of these fibrous proteins. They take on an alpha helix configuration, and then three of those alpha helices will wrap up together. So this forms a very strong fibrous protein. Okay, hemoglobin. Hemoglobin consists of 4 different globular polypeptides. Okay, so both of these have quaternary structure. But there's another way to have quaternary structure, and that's through the addition of prosthetic groups. A prosthetic group is part of a protein that is not proteinaceous, but is required for it to function properly. So what does non-proteinaceous mean? It means it's not made of protein. Well, what does that mean? It means that it's not made of amino acids. Okay? So prosthetic groups can be either organic, like this retinol down here. Retinol is a lipid. So look at the lack of polar groups here. And retinol is the prosthetic group of the protein rhodopsin. This is rhodopsin. And rhodopsin is a protein in your eye, and it senses light. And the way it senses light is that this prosthetic group, retinol, changes its configuration when it is excited by light. So when there is no light, it looks like this, where it has this cis bond, this cis double bond here. When it is excited by light, the cis bond turns into a trans bond. Now you have a straight chain. Without retinol, rhodopsin doesn't function. You cannot sense light without the changing of that lipid. But it's not proteinaceous. It's not made out of amino acids. Okay? So another prosthetic group is in this hemoglobin. You have these heme groups. Now, heme groups are not organic, right? This is organic. It's made of carbons and hydrogens. But heme consists of a metal, consists of iron. And so heme groups are not organic prosthetic groups. They're not made of amino acids, so they're non-proteinaceous. They're an iron ion that's surrounded by an organic ring. And it's that iron that is going to be responsible for binding to oxygen. And so without that heme group, hemoglobin can't move oxygen through your blood. And so hemoglobin can't function without its prosthetic group. It's not proteinaceous, but it's required for the proper functioning of the hemoglobin. All right, so here we have collagen, rhodopsin, and hemoglobin. All of these are proteins with quaternary structure. Collagen has multiple subunits but no prosthetic groups. Rhodopsin is only a single subunit. This is a single polypeptide, but it has a prosthetic group, the retinol. And hemoglobin has both. Hemoglobin has 4 different subunits, 4 polypeptides, and it also has a prosthetic group. Okay, so you can do that in different ways. All right, what I would like you to do, what I would like you to do at this point is to think of a protein, find a protein. It can be a toxin, it can be something that functions in your body, it can be a structural protein, I don't care what it is. And I want you to characterize it. Okay, I want you to characterize its 4 levels of structure. All right, so all right, I'm not going to talk too much about this. These are things that you should know. I'll let you study diseases due to protein structure on your own. And I also want you to go through these vocabulary terms. Okay, so motif is smaller than a domain. A family has to do with evolutionary history. A complex is unrelated proteins that all come together to do a single function. An assemblage is repetitive subunits. So I want you guys to go over that stuff on your own. And then finally, I'm going to talk about 2 different ways to activate and deactivate proteins. Okay, because what the cell will often do is have a certain suite of proteins available in the cytoplasm. But whether or not those are turned on or off will determine whether they're functioning or how they're functioning. Okay, so one common way to activate and deactivate proteins is by phosphorylation. Okay, so by attaching a phosphate group to a protein, you change its conformation, and that will either turn it on or turn it off. Okay, and this is cyclic. Oftentimes, ATP will be used as the substrate where you're getting the phosphate, but sometimes that phosphate will come from another protein. Okay, so in this case, in this cartoon, you have ATP is being hydrolyzed to ADP, and the phosphate is being added to this protein. Now this protein is activated, it goes and does stuff, and then you have a phosphatase which comes and removes that phosphate, and now you have the inactive form. Okay? Another common way is the GTP binding cycle. Now, proteins that are GTP binding proteins will be active when they are bound to GTP, and they will be inactive when they're bound to GDP. Okay? So there'll be a molecule that activates these proteins by encouraging, it'll change the conformation of the protein and encourage the GDP in the binding site to leave. So the GDP will be bound in the nucleotide binding site, and when the protein will be activated, the GDP molecule leaves the protein, and a new molecule of GTP from the cytoplasm will enter the nucleotide binding site. Okay? Don't get this mixed up. A lot of people want to say GDP is phosphorylated to GTP. That doesn't happen because that is an energetically expensive process, right? GTP is almost the same thing as ATP, and proteins can't just make ATP willy-nilly, right? You have to go through respiration to make ATP. The same with GTP. So GTP is being formed in processes similar to cellular respiration. And so you have a GTP pool in the cytoplasm. So these proteins will get rid of the GDP that is bound, and they will attract a new molecule of GTP in order to become activated. All right, so here's a nice little cycle. There'll be a signal, and that will cause what's known as GEF activity. GEF stands for guanine exchange factor. There's lots of these, and we'll talk about specific instances in the semester. So guanine exchange factor stimulates this GTP binding protein to get rid of the GDP. This molecule of GDP leaves the protein, and a new molecule of GTP enters the protein. Okay, now it's active. Now, the protein itself, usually, sometimes something else has to work on it, but most of the time, these GTP binding proteins have intrinsic GTPase activity. A GTPase means that it's an enzyme that hydrolyzes GTP. So intrinsic means that the protein has it itself. Okay, so the protein will hydrolyze the GTP to form GDP, right? That's an exergonic process that releases energy. So that's not a problem. It's not a problem to hydrolyze the GTP. It's not possible for these proteins to phosphorylate a GDP. That's an energy expensive process that requires things like cellular respiration. All right, so you have an inactive protein. A signal comes in the form of guanine exchange factor that causes the GTP binding protein to get rid of the GDP and to accept a new molecule GTP. When it's bound to GTP, then it is in its active form. Most of these proteins only stay active for a short while. That's not true for all of them, but many of them only stay active for a short while. So if you want to continue the signal or continue the activity of the protein, you have to continuously reactivate it. Because they have an intrinsic GTPase, which after a short while will cleave the GTP, will hydrolyze the GTP, removing a phosphate, and resulting in the protein being bound to GDP, which is the inactive state. All right, so why don't you answer that? All right, so, all right, at this time, I would like you to go to the TED Talk that I've posted on our website. It's about spider silk proteins. And I'd like you to watch that TED Talk. And then when you're done with the TED Talk, I'd like you to find another protein that is being used for some sort of human benefit. Okay? It doesn't necessarily have to be biomimicry, although I encourage you to find something that's being developed as a biomimicry protein. But if you would prefer to find, a drug or some other protein, it must be a protein. And I want you to characterize it. Now, I want this one to be different from the one that you characterized in terms of its structure, primary, secondary, tertiary, quaternary. This one, instead, I want you to focus on the other terms. So, are there different protein motifs? What are the protein domains that are the functional domains? Is it part of a protein family? Okay, so I want you to use those terms in order to describe your protein. Okay, so for this lecture, what I need from you for follow-up assignment is 2 different proteins. One of them you're characterizing in terms of structure, primary, secondary, tertiary, and quaternary if it has it. If it doesn't, you need to say that. And a different protein that is being used, that's being developed for human use. Okay. If you want to do a biomimicry protein, I encourage that, but I won't take off points if you don't. And that protein I want you to describe in terms of the other vocabulary that are in this lecture. Protein motif, protein domain, protein family, protein complex, protein assemblage, if any of those terms apply. Okay? All right. I will see you guys the first week of December. Don't forget, you need to do your assignment for lab, which is to describe 5 different types of media. And then once you've done that, if you want to go ahead and get started on making 3 of those media, that would be fine. All right, I will talk to you later. Let me know if you have any questions.